2010, Siberia. Russian scientists made a discovery that shouldn't exist. A woolly mammoth calf, 42,000 years old. But this wasn't just bones. It was tissue, muscle, skin, internal organs, all intact. Her stomach still contained her mother's milk. And when they examined her cells, they found DNA, not fragments, complete sequences. 42,000-year-old genetic code, still readable. Here's what makes no sense. Meat in your freezer gets freezer burn in weeks. DNA in biological samples degrades in months, even frozen. So how did glacial ice preserve tissue for 40,000 years, better than modern technology can preserve it for 40 days? The answer is physics, very specific physics. And it has nothing to do with cold alone. Water is constant motion. Molecules vibrating, rotating, colliding. At room temperature, each water molecule moves at about 1,500 miles per hour. That motion is heat. When you cool water below 32 degrees Fahrenheit, the molecules slow down, stop moving past each other, lock into position. Ice. But here's the critical detail. Ice takes up more space than water. About 9% more. When water freezes, it expands. And if that water is inside a cell, the ice crystals grow like knives, puncturing the cell membrane, shredding DNA, destroying the delicate machinery of life. This is why freezing damages tissue, not the cold itself, but the ice. But not all ice is the same. The speed of freezing matters. How the ice forms matters. Fast freezing creates millions of tiny crystals. Slow freezing creates fewer, larger crystals. In Siberian permafrost, freezing happened slowly, over weeks, months. And critically, once frozen, the temperature never changed. -20 degrees Celsius for 40,000 years. This slow, stable freezing did something remarkable. Instead of shredding DNA, the ice crystals grew around it, gently displacing the molecules, encasing them. The DNA ended up suspended in microscopic pockets, protected from oxygen, protected from bacteria, protected from degradation. Perfectly preserved. Your freezer is terrible at preservation. Not because it's not cold enough, but because it's unstable. Every time you open the freezer door, warm air enters, temperature rises slightly, some ice melts. Then, when you close the door, it refreezes. This cycle happens hundreds of times. Each time, ice melts and refreezes. New crystals form in different patterns. Cell membranes tear a little more. DNA strands break. Within weeks, the tissue is destroyed at a molecular level. This is freezer burn, not burning from cold, but damage from instability. Permafrost doesn't fluctuate. -20 degrees in the year 40,000 BCE, -20 degrees in the year 2024 CE. Perfect stability. The top few meters of ground thaw in summer, but below that, permafrost. Ground that hasn't thawed since the Ice Age, hundreds of meters deep, a natural deep freezer, perfectly calibrated, perfectly stable. When mammoths died in these conditions, if they fell into cold mud or water, and if winter came quickly, they froze slowly but completely, before bacteria could decompose them, before oxygen could oxidize their tissue. And then they stayed frozen for millennia. When scientists extract tissue from frozen mammoths, they work in sterile rooms cooled to -20 degrees. Because thawing is the most dangerous moment. Inside the cells, they found structures that shouldn't survive. Mitochondria, the cell's power plants, ribosomes, the protein factories, even nuclei containing the chromosomes. All intact, recognizable, as if the mammoth had died yesterday. The DNA was extraordinary. Not just fragments, not just scattered base pairs. Long sequences, complete genes, enough to read the mammoth's entire genome. In normal conditions, even refrigerated, DNA degrades through hydrolysis. Water molecules slowly break the bonds. Half-life of about 500 years. But in ice, the water is locked solid, can't interact with DNA, can't break it down. The half-life extends to hundreds of thousands of years. Scientists have now extracted readable DNA from specimens over 1 million years old, frozen in permafrost, still intact enough to sequence. This is time travel, molecular time travel. But DNA isn't all they found. One mammoth, a male named Yucca, still had liquid blood in his veins, 40,000-year-old hemoglobin. Still red, still chemically functional. Researchers extracted it, analyzed it, and found it was adapted for extreme cold, a hemoglobin variant that released oxygen more efficiently at low temperatures. They were reading the molecular adaptations of an extinct species from preserved blood. Today, companies offer cryogenic preservation. Freeze bodies in liquid nitrogen. -196 degrees, hoping future technology can revive them. But they're fighting the same problem. Modern freezing is fast. Too fast. Ice crystals form rapidly, creating the same cellular damage. Unless they use cryoprotectants, chemicals that prevent ice formation, turning tissue into glass instead of ice. This is called vitrification. Freezing without crystals, preserving the exact molecular arrangement. It works, but it requires chemicals mammoths didn't have. Nature achieved near-perfect preservation through patience, through stability, through 40,000 years of unwavering cold. We're trying to do the same thing in an instant. But the permafrost is melting. As the climate warms, the natural freezers are failing. Mammoths that stayed frozen for 40 millennia are thawing, decomposing. Their DNA, preserved for longer than human civilization has existed, is degrading in years. Scientists are racing to extract and catalogue as many specimens as possible before they're lost. The ice that preserved them is the same ice whose melting will destroy them. For 40,000 years, ice was the perfect preserver. Stable, patient, eternal. But nothing is truly eternal. Not even ice.